Hello, good afternoon everybody. My name is Saurabh Shawkar and I am a biology tutor. I have done my masters in human physiology. So right now I am going to discuss the portion of digestion, that is the part of life processes, mainly the part of nutrition. So in case of the life processes, the main major life processes are reproduction, then excretion, nutrition, and then there is you can say excretion. Okay, and in the nutrition portion they have different steps. Okay, the first one is ingestion, then second one there is digestion, after that assimilation, absorption, assimilation, and after that there is excretion. So all of these portions are there in case of the nutrition, and right now I am going to talk about the steps of digestion, mostly how juices are going to help to break down complex food product complex food product into the simple one, simple food. Okay, now before that we have to understand, or we already know what are the components of food. So what are the components of food? There is carbohydrate, protein, fat, right? And after that there is minerals, vitamin and roughage. Okay, so all those things are there, but mostly carbohydrate, protein, fat, they are going through the process of digestion. Now you can see there are chemical known as enzyme. Okay, so enzymes are what? Enzymes are the protein in nature, sorry, protein in nature component, and which act as a biocatalyst. What is the meaning of catalyst? Catalysts are the component, or catalysts are the chemical substance that control the rate of reaction, or you can say that initiate the reaction. Okay, and after the reaction they will stay unchanged. After the reaction they will stay unchanged. So enzymes helps in control the breakdown of any kind of food or food components, and they also initiate in the digestion of or breaking down of any kind of food products, and after the reaction they will stay unchanged. Okay, and why they are bio? Because enzymes are mostly found in biological components, or you can say living component or biotic component. Enzymes are present in biotic component, so that's why they are known as biocatalyst. Okay, now specificity related to enzyme, there is a theory known as lock and key theory. What is the meaning of lock and key theory? Because for each lock there is a separate key. Without that particular key we can't open the lock, right? Mostly without that particular key we can't open the lock. So in case of enzyme, for a specific substrate there is specific enzyme. Okay, so just like if you say if this is one enzyme, and just think about, this is the active site, which substrate is going to fit in that portion on that active site, that substrate will react with that particular enzyme. Okay, so if this is one enzyme and in that specific portion another substrate can be fit. Okay, and when the particular substrate with particular enzyme get attached, that will make enzyme substrate complex. Enzyme will break down that substrate. Okay, enzyme will break down that substrate into but different components, and that will make enzyme product complex, and after that enzyme will stay unchanged. Enzyme will stay unchanged with that active site, but the product that will come out after the reaction, these are product. Okay, so first substrate attached with the specific enzyme, specific substrate will be attached with the specific enzyme that will make a substrate complex. After that the substrate will get changed into product, and that will make enzyme product complex, and after that the product will come out from the active site of the enzyme, and the enzyme will stay unchanged. That is specificity. Okay, not all you can say all subject are not going to, or substrate, all the substrate are not going to attach with all the enzymes. For specific substrate there is specific enzyme. Now optimum environment for the enzyme, they require optimum pH. Okay, some enzymes are going to activate themselves in acidic pH, some are going to activate themselves in the neutral pH. Okay, and there is another thing, that is optimum temperature. Optimum temperature means, so in between 25 to 40 degrees centigrade the enzymes are mostly functionable. Okay, and they can show their higher amount of activity. And if the temperature is more than 40, or if the temperature more than, you can say 80, the temperature is more than 80, or if the temperature is less than 0, temperature is less than 0, then the enzyme can't do their function. Enzyme can't do their function. Okay, understood. So enzyme mostly stay activated in between 25 to 40 degrees centigrade. And is the enzyme is in the 25 degrees centigrade, for each 10 degrees centigrade, so 25 to 35 the enzyme activity will increase, 35 to 40 enzyme activity will again increase, but above 80 degrees centigrade that can't perform properly. Okay, so each enzyme they have a optimum temperature level. Okay, so these are the characters of enzyme. Enzymes are protein in nature, and we know proteins are made up of amino acid, right? So we can say the enzymes are made up of amino acid, or we can also say enzymes are made uh, indirectly or directly by the function of ribosome, right? Because ribosome helps in protein synthesis. Okay, so now next portion, that is its digestive system. Now here you can see the picture. Okay, and if you draw the picture, so in this portion there is buccal cavity, right? In this portion there is buccal cavity. Now in this portion there will be nasal tract. So nasal tract and buccal cavity, they are going to attached in this portion. This portion is known as buccal cavity. Okay. Now in the buccal cavity portion they have salivary gland. They have salivary gland. How many salivary glands are there? Total 3 pairs of salivary gland. 3 pairs of salivary glands are there. Okay, what are the name of those salivary gland? Let me write. Okay, sorry. What are the name of those salivary gland? The first one, that is parotid. That is the largest salivary gland that will stay in this portion. In this portion, here you can say parotid. Okay, the second one, that is known as sublingual. Okay, and third one, submandibular or submaxillary. Okay, these three, sublingual, submandibular and parotid, these three salivary glands are there. This salivary gland release the enzyme that is known as salivary amylase or dialin. Salivary amylase or dialin. Okay, and another component, chemical component, that is known as lysozyme. Okay, These two components are released by the saliva. Now salivary amylase or ptyalin, they break down the carbohydrate. They break down carbohydrate. So we can say from the complex carbohydrate, they break down complex carbohydrate into the simpler one, simple carbs or simple carbohydrate. So what are the complex carbohydrate? Okay. So complex carbohydrate means that could be polysaccharide. Polysaccharides means poly means more than two, okay, or more than one equals. Polysaccharide means if there is a long chain of carbohydrate, okay, just like starch, or you can say glycogen. So they are a long chain of glucose. Long chain of glucose are making those structure. So they are complex carbohydrate. Now there is disaccharide. Disaccharide means di means two. Okay. So if there is the uh, monosaccharide, glucose plus glucose, that will make maltose. Okay. Glucose plus fructose, that will make sucrose, and glucose plus galactose, that will make lactose. These are all disaccharide because there are two sugar molecules. Okay. Now the simple sugar, that is known as monosaccharide. Simple sugar, only one glucose molecule is there. Only one sugar molecule is there. Just like glucose, then fructose or galactose. Okay. So these are the simple sugar. So from the complex carbohydrate, from the polysaccharide or from the disaccharide, they will be broken down to the monosaccharide by the help of you can say digestive juices, or you can say enzyme. Okay, by the help of enzyme. Acha, if there is any question, which bonds the digestive juices are breaking the bonds in between two, or breaking the bonds of long chain, that's why they are going for the simple one, right? We have to break the bonds and then we can get the simple or the basic product. So in between the glucose molecule, they have glycosidic bond. Glycosidic bond. Okay. So in each step we are breaking down glycosidic bond and we are getting the simplest sugar. So in case of salivary amylase, that is also known as ptyalin, is breaking down the starch or glycogen into maltose. Okay, the starch or glycogen, okay, they are going to broken down into maltose. Okay. Then after that, in case of lysozyme, also talked about lysozyme. What is the function of lysozyme? It is not a digestive juice, but it is a chemical that kill microorganism, that helps in cleaning the kill the microorganism. Okay, so that is the portion in buccal cavity by the juices. Now this is the chemical digestion. This is known as chemical digestion in the buccal cavity. Okay. Chemical digestion means where chemicals means enzymes is helping in breaking down of the food. In the mouth or buccal cavity, we can also see mechanical digestion. What is the mechanism of mechanical digestion? Where we are using muscle or any kind of parts of our body, or any kind of organ to break down the food without using any chemical. Just like in case of the mouth, teeth cut the food, chew the food, and from the large food product, they are converting into the simpler one, right? So teeth are not using any kind of the chemical, right? They are physically by muscular energy, they are breaking down the food without using the chemical components, right? So teeth are showing mechanical digestion. Now, in case of tongue, what is the function of tongue? The tongue helps in mastication. Okay. Mastication means mixing of food with saliva. Mixing of food with saliva. Okay. So that the food can easily dissolve or get absorbed the saliva and easily absorb the saliva, and they can be easily broken down. Okay. Mastication. The second one, tongue also helps in getting the taste of the food, that enhance the releasing of saliva if there is the food is tasty. Okay, that will be controlled by our nervous system, or that will send some signal to the brain, and the salivary gland will release more amount of saliva, right? So tongue is helping in taste, getting the taste, and also helps in swallowing. Okay, also helps in swallowing. So these are the process that is shown by the tongue or shown by the buccal cavity portion. Now, which food, after breaking down the food, mixing the food with the saliva, and not all the starch or carbohydrate is going to break down, some amount of starch or carbohydrate will be broken down in this portion. Okay, and that will convert into the maltose. Okay, so after partial digestion, we will not say complete digestion of carbohydrate, we'll say partial digestion of carbohydrate. Okay, that mixed component, the food with the saliva, that mixed substrate is known as bolus. Now that will enter into the esophagus, the long tube-like structure from the buccal cavity up to the stomach. So from the buccal cavity up to the stomach. Just like this. Okay, and to the stomach, this portion is known as esophagus. Now esophagus, so esophagus do not show any kind of digestion, or esophagus do not release any kind of enzyme, but they show one kind of movement known as peristalsis movements. Peristalsis movement. Okay, so peristalsis movement helps in moving the food forward, moving food forward in alimentary canal. Okay, moving food forward to the alimentary canal. So alimentary canal means what? From the buccal cavity, okay, up to the anus. The whole tube-like structure or tubular portion is known as alimentary canal. Okay, so in the alimentary canal, the food move forward by the process of peristalsis. Okay. Now, in case of buccal cavity, protein or fat, they are not going to digest. Okay, only carbohydrate and partial amount of carbohydrate is going to be digested. Now, when the food will enter, or when the bolus that will enter into the stomach, that will get absorbed, or that will start reacting with HCl. Now, in the stomach, okay, there are three type of cell. One is peptic cell. The second one is oxyntic or parietal cell, and the third one, that is known as mucous cell. Okay, this three. Achha, in case of buccal cavity, I have said na, each of the enzyme required some optimum pH. So in case of salivary amylase, they require the pH near about you can say 6.7. Okay, if the pH is lower than 4, if the pH is lower than 4, then salivary amylase can't cause any kind of digestion. Okay, salivary amylase will stay inactive. Okay, only pH near about 6.7 it can show some function. But in case of stomach, the stomach enzyme or enzymes in the stomach, they only get activated when the pH is near about 2 to 3.7. The pH is 2 to 3.7. Now in case of stomach, the peptic cell, they release pepsinogen, and you can say prorenin. Okay, pepsinogen and prorenin, these are the two enzymes. Okay, and pepsinogen and prorenin, these are the inactive form of enzyme. These are the inactive form of enzyme. So who will active them? HCl. The parietal cell release HCl. So pepsinogen is going to activated by HCl, and that will create pepsin. Okay, prorenin is going to get react by HCl, and that will make renin. Okay. Now what is the function of mucus cell? That will release mucus. Okay. So what is the function of HCl? If you say activate the enzyme, activate enzyme, right? And the second one of the function of HCl, that can kill microorganisms, because in case of street food or in case of any kind of unhygienic food, somehow we have taken, okay, in our body, then there should be some barrier that will protect our body from all of those microorganisms, right? So the HCl will kill the microorganism. First in this portion there is lysozyme, and then near to stomach, in the stomach there will be HCl that will kill the microorganism. Mucus is a fluid like texture, okay, that helps in uh, that helps the stomach wall from getting damaged by the action of HCl. Okay, so mucus will create a layer. There will be a layer of mucus, okay, so that the HCl do not get direct contact, because the pH is too low and the pH is too acidic. Okay, if the HCl gets direct connection with the wall, then the stomach wall could get damaged. Okay, so that's why to protect stomach wall from the HCl, there will be a layer in between them, that is mucus layer, and mucus is released by mucus cell. Now there will be question that what is the function of pepsin and renin? Now renin will react on the milk protein, that is known as casein. That is milk protein. So in case of in case of baby, or in case of, you can say, infant, okay, who are mostly depend on their mother's milk, okay, they, in that case, their body will release one kind of their stomach will release the enzyme renin, that will break down the milk protein casein into paracasein, and paracasein is the easier form the body could absorb. Okay, now in case of pepsin, okay, so renin could found in case of baby or in case of infant, but in case of adult, renin is absent. But in case of pepsin, pepsin will break down the protein into peptone. Okay, protein to peptone, or you can say peptide. Okay, so this is the function of pepsin. Okay, because if you see the structure of protein, protein is made up of protein is made up of peptides. Okay, peptides are made up of amino acid. Okay, so if there is amino acid, they are making connection to one another, that will make peptide, and one peptide will get joined by another peptide, and that will make whole protein. So the simplest form of protein, that will be amino acid, but in the stomach we are not getting the amino acids directly, we are getting peptide in the stomach. Okay, so here you can say in case of stomach there is partial digestion of protein. Okay, understood. Now, when the saliva was mixed with the food, the partially digested food was known as bolus. When the food is released by the stomach, the acidic food or partially digested acidic food is known as chyme. Okay, so bolus, then chyme. Now the chyme will enter into the small intestine. So if I draw, that is duodenum. In this portion there will be liver. Okay, okay. In the side of stomach there will be liver. Okay, and liver is important in case of digestion because liver produce a chemical component known as bile. Okay, and bile will get stored in gallbladder. Bile will get stored in gallbladder. Now what is the importance of bile? From the stomach the chyme will enter, and the pH of chyme is near about 2 to 3.7, but the juices or what the gastric juice that is released by pancreas, that will be which will be activate only in alkaline pH, that will be near about 8.4. When the pH is 8.4, then the enzyme from the pancreas can do their function. So to reduce the pH, that will be done by bile, the first. Second, the bile helps in emulsification of fat. Emulsification of fat. What is emulsification fat? Now we know that fat are not easily dissolve, or not is fat do not get dissolved in the water, right? As the fat will not get dissolved in the water, it is quite tough to digest the fat. Okay, so if there is a large globule of fat, the bile component, the bile salts, okay, the bile salts are sodium bicarbonate, then sodium glycocholate, sodium taurocholate. Okay, all those bile salts will break down the larger globule of fat into the smaller globule. Okay, now here you can see the surface area of the larger globule is not so much, but when it is broken down in the smaller droplet, the surface area is increased. The surface area is increased. Now easily the gastric juices from the intestine and the pancreas, they can react on the fat droplet and they can easily digest the fat globules. So breaking down the larger globule into the smaller one is done by the bile, that is emulsification of fat. Now the third one, that is there are some vitamin that is fat soluble vitamin. Okay, that is ADEK. All those vitamins are also absorbed to the body by the help of bile. Okay, so bile has three functions: one, reducing the pH; two, fat soluble vitamin, absorbing fat soluble vitamin; and third, emulsification of fat. Okay, now the second one, that pancreas. Now what is pancreas? Yes, in case of liver, that is the largest digestive gland, largest internal gland or largest digestive gland. Okay, digestive gland. Okay, now pancreas. pancreas is known as mixed or heterocrine gland. Okay. Now, what is the meaning of mixed or heterocrine? Hetero means different, just like heterotroph. Troph means nutrition, heterotroph different nutrition. Heterocrine. Okay. So when any gland showing more than one function or related to more than one function, those glands are known as heterocrine gland. So pancreas act as exocrine, where they release the enzyme. Okay, or you can say pancreatic juices. And they also act as endocrine, where they release hormone. Hormone means you can say insulin, then there is glucagon, somatostatin. Okay, we'll talk about those things in the endocrine chapters. Okay, mainly control and coordination. Okay, all of these things are there. Now right now we are talking about exocrine function. Okay, so exocrine means enzyme. So which enzymes are there? So in the pancreas they will release trypsinogen and you can say pancreatic lipase and pancreatic amylase. Now trypsinogen is the inactivate form. How trypsinogen will get active? When the bile salt will reduce the pH. Okay, when the bile salt will reduce the pH and turn the pH at 6 near about 8, turn the pH at near about 8, trypsinogen will convert it by an enzyme known as enterokinase. Okay, enterokinase into trypsin. Okay, trypsin. Now what is the function of trypsin? Trypsin will break down the peptone. In case of in the stomach, protein will was broken down to the peptone. Now the peptone or peptide will be broken down to the amino acid. Understood? Okay, so amino acids, they are connected by the peptide bond. We broke down the peptide bonds and got the simpler form of the protein, that is amino acid. Okay, so complete digestion of the protein has been done by the help of pancreatic juices and in the position of duodenum, where it is happening. So all these juices, bile and all these juices, okay, they will be released in the intestine, the duodenum of small intestine. Okay, so in the duodenum the complete digestion of protein has been done. Now it will be absorbed. Okay, second, pancreatic lipase will break down the fat globules in fatty acid and glycerol. Okay, now fat globules in between in the fat, the components of fat are connected with one another by the name of ester bond. Okay, so in case of protein they have peptide, in case of fat they have ester bond. We have to break down the ester bond, we will get fatty acid and glycerol. The pancreatic lipase will do that. Okay, so digestion of fat is done in the pancreas portion or in the small intestine portion, sorry, in the small intestine portion, in the duodenum portion. Okay, now pancreatic amylase. So in case of carbohydrate, they are connected by which bond? They are connected by glycosidic bond. Now in the mouth, the partial digestion of carbohydrate was done, right? The glycosidic bond, but for few particles or for few starch and few glycogen, the glycosidic bond was broken down and we get maltose. Now maltose is a disaccharide. We have to break down into monosaccharide Monosaccharide is the simplest form. So the maltose, or you can say sucrose, or you can say lactose, okay, maltose, sucrose, or lactose, they will broken down, sucrose or lactose, they will be broken down to glucose, mostly converted to glucose, okay, and then the body will absorb, okay, all those things, okay, the end product will be glucose. So these are the things. So pancreatic amylase, that will helps in breakdown of carbohydrate, pancreatic lipase, that will helps in breakdown of fat, and trypsinogen, that is inactivate form of enzyme, that will get activate and that will break down partially digested protein, that is peptide or peptone, into amino acid, okay. So these are the portion of complete digestion of you can say complete digestion by digestive juices, okay. So let's revise the portion what we have learned. We have learned that there are different steps of nutrition, okay, and the digestion is the state where the complex food product will be broken down into the simpler one, okay. Now in case of enzyme, they are mostly protein, they are biocatalyzed, control or initiate the rate of reaction, okay. They remain unchanged after the reaction, okay, means they can be used in another time. So specificity, for specific substrate there is specific enzyme, optimum environment for each enzyme, they require optimum they require specific pH, right, optimum pH, we already get those ideas, okay. Just like in case of trypsin they write alkaline pH, in case of pepsin they required acidic pH. Then optimum temperature, okay. Then we learned about parotid, sublingual and submandibular gland, which is salivary gland is releasing salivary amylase or ptyalin, breaking down complex carbohydrate into the simpler one, okay. Then we talked about different type of complex carbohydrate, polysaccharide and disaccharide, and the simplest form of the carbohydrate, that is monosaccharide, glucose, fructose and galactose. Now chemical digestion is the way where the digestion is been done by chemicals, and in case of mechanical digestion, chemicals are not being used, or the enzymes are not being used. Instead of enzyme, the muscular power or any kind of organ will break them down without use of enzyme or without use of any chemical. That is mechanical digestion. Mastication, mixing the food with the saliva by the help of tongue, okay, that is mastication. Bolus, the component that mixed with saliva released by the buccal cavity and that entered into the esophagus, okay. We learned about the peristalsis movement. In the stomach, there are three cells: peptic, parietal, and mucous cell. Peptic cell will release pepsinogen and prorenin. Pepsinogen will create pepsin, and prorenin will create the renin. Okay, renin, that is only for infant. Okay, and not for adult, and pepsin for adult and infant both. Okay. Now protein, that will be broken down by pepsin into peptone and peptide. Okay, so proteins will be broken down in peptide, and peptide will be broken down in amino acid, but that is not being done in stomach. In stomach, the protein only broken down to peptide. Peptide amino acid conversion will be done in duodenum or small intestine. Okay. Now the largest gland, that is liver, release bile, and bile will be get stored. Bile will get stored in gallbladder. Now if there is any question why bile is being stored, because if we are continuously taking fatty food, the liver has the liver can't make that amount of bile. Okay, the formation of bile is too slow. Okay, so liver have to store them whenever the fat, whenever we are eating any kind of fatty component, fatty food component, then at that time, okay, the bile will release. Okay, so as the formation of bile is too slow, we have to store them. Only when it is required, it will be released. Okay, now there is a duct known as hepatopancreatic duct. Hepatopancreatic duct means the duct that is coming from the liver. Okay, the duct that is coming from the pancreas, they will join together and make hepatopancreatic duct. Hepatopancreatic duct, and through which the pancreatic juices and bile both will be released to the duodenum. Okay, then we will talk about trypsinogen, pancreatic lipase, and pancreatic amylase. Okay, so digestion is done. Now the next day we will talk about the absorption, how the absorption is happening mostly in the small intestine and large intestine portion. Okay, so digestion will be completed in the small intestine portion. Okay, I hope there is no doubt, but if there is any doubt, please feel free to ask in the next class. Okay, so right now, bye bye, see you in the next class, take care.